Okay, good afternoon, everyone. First, I want to thank GLBIO for giving me an opportunity to present a talk. And in addition, I'm grateful to the NSF Travel Fellowship as well. So I'll get into it. On a high level, our lab is focused on developing informatics methods to analyze gene regulation and transcriptome data at the isoform level. By this, what I mean is that different transcript variants are driven by alternative transcription and alternative splicing, which lead to different protein isoforms from the same gene. So based on current genome annotations, about 50,000 genes, which include both protein coding and non-coding, transcribe around 200,000 transcript variants, leading to about 100,000 protein isoforms and 100,000 non-coding variants, implying that genes have two or more protein isoforms. So understanding this highly complex process in normal tissues and what goes wrong in disease scenarios is one of the main objectives of my lab's research. Our lab in the past, before I joined, and others have published a series of papers showing the limitation of gene level analysis and emphasizing the importance of treating isoform or transcript variant as a unit of measuring, measure rather than gene as a unit of measure in various computational analyses.
Sorry to interrupt. Slides aren't sharing online. I just need to tweak this real quick. Okay, you're good. Sorry.
Thank you. For example, opposing expression patterns of transcript variants during brain development are observed, and then isoform level analysis can better stratify tumor and non-tumor patients from cell lines or even bulk RNA-seq. And isoform level analysis also shows better association with clinical outcomes like survival. Okay. So next I'll talk about tissue specificity. All human cells carry out common processes in their tissue environment, yet each tissue has unique functions which define their particular phenotype. So these isoforms have been shown to influence gene expression through post-transcriptional changes and epigenetic mechanisms like splicing, and this often happens in a tissue-specific manner. So we explored this idea using GTEx, which is normal tissues dataset, which provides a rich resource to understand these complex regulatory patterns, which are tissue specific, and thereby eventually will assist us in understanding drug target interactions, biomarker discovery, prognosis in complex diseases. Now, our lab was the first one to develop a genomic language model called DNA-BERT, which is used for unraveling the language of non-coding regions. So when I started out, I wanted to apply that to kind of understand tissue-specific language among promoters and other splice regions. But when we started preparing the training data, we found that there is no gold standard of tissue-specific transcripts, for example. And then there is a lack of consistency across the methods, as shown in the graph, where two different scores like tau and z-score give very different outcomes. So that brings me to the objectives of this paper, where we want to classify transcripts into different expression groups based on their expression patterns in normal tissue. And then we want to validate it across different methods and understand their features like biotypes. And the next is to identify the driver of tissue specificity, and in the end, I want to talk about brain-specific transcripts and what happens to them in disease. So eventually, the goal is to use these transcript sets in classifiers for patient subgrouping, which would be clinically useful and cost-effective, and also to use it in DNA-BERT fine-tuned models to understand their language. So that brings me to the scoring method. So if you can see, okay. So in the box there, in the gene expression matrix, I'm comparing lung samples with all the other samples, all the other tissues. And for a given transcript one, we want to compare it with samples in lung versus the rest. So first, what we do is we define the probability of the jth transcript being expressed in the ith tissue as Pij, which is the number of samples with an expression value greater than 0.5, which is a threshold used overall, divided by the total number of samples in the ith tissue. And the next thing I calculate is the inflection point, which is basically the significant change in probability of expressed transcripts. So once this is done, it classifies it into tissue-specific, tissue-enhanced, and widespread on the right side, which is the probability of high expression, and on the left side into null and low groups. The second calculation is the fold change max calculation, where basically you calculate fold change by comparing with the max of the rest of the means. So this basically helps in separating out very closely related expression patterns. And finally, we compute the empirical p-values from stratified subsampling approach, which my lab published in 2021. So what we see is that by applying this method to the transcript level expression profile, we take about 10,000 samples, which is based on the RNA integrity value of 7 across 26 major tissue groups. We found that 92,000 of them, or 46% of the total transcripts, which correspond to 27,000 genes, both protein coding and non-coding, have a probability greater than the inflection point for at least one tissue. And the probability where it's greater than the inflection point in exactly one tissue is about 25% of the total transcripts. So according to these groupings, what we see here in this in the groups diagram is that only 9% of the total transcripts contribute to tissue specific, and a majority of them are classified into either null or low. And among the tissue specific transcripts, we have testes, which has the highest number, and many studies have reported this. So this wasn't a surprise, followed by liver, brain, pituitary, and muscle. So the next thing we wanted to do, since there is no gold standard, is to compare with other methods and see what they've classified as tissue-specific versus not. So we compared with SRTDB, where they classify tissue-specific transcripts from cancer and normal tissues, and they find out 44,000 of them. And then we also do a tau score calculation, which is kind of like a widely used method for tissue-specific calculations at a 0.85 threshold. And surprisingly, only 4% of the total transcripts, which is the 2,000 number in the center, are categorized as tissue-specific in consensus by all the methods. So to further validate this, I next compared it with gene-level analysis, although this is not an apple-to-apple comparison, because they are experimentally validated across the single and the sum category of human protein atlas. And an interesting finding here is that although our method shows good concordance as compared to the other two methods, we see that majority of the tissue-specific genes of the tissue-specific transcripts are either tissue-specific in one tissue, and the other transcripts of that gene are either null or low in expression. So this suggests that only one transcript of a gene, which is highly tissue-specific, is really necessary for that given tissue of interest. Okay. To further understand the transcript length and biotypes, we see that the transcripts expressed in multiple tissues are longer than the ones which are either specific or null or low. And also that retained introns are most common in genes expressed in tissue-specific low or null. So basically, they're very specific to a tissue or they're not expressed at all as compared to those expressed across multiple tissues. But then we wanted to see what's driving tissue specificity with the information of the expression matrix that we had. So we looked, what we found here was that majority of the genes have only one transcript per gene, which are tissue specific, and the rest, which is 33%, are multi-transcripts. So many transcripts of a gene are tissue specific. So one interesting thing is that among these multi-transcript and multi-tissue ones, which are 11%, we see that alternative tissue-specific transcripts are in a majority, where basically, if you look at the box on the right side, where the same tissue and the different promoter category. So the conclusion here is that higher number of multi-transcript tissue-specific genes in the multi-tissue category are likely to use alternative promoters as in compared to the other class. So tissue preference can describe alternative promoter activity, although this is only based on the expression data. But then to really understand if this is happening, we try to look at the cell types enriched in the brain-specific transcripts. Of course, we mapped them to the genes and then looked at the cell types enriched. So what we found was that out of the 251 out of the 436 brain-specific transcript genes were annotated as cell type markers in a database called scBrainMap, which came out about a year ago. And majority of these genes are annotated in the non-neuronal subtypes in the diagram on the right, where mostly in astrocytes, oligodendrocytes, etc. And as per annotations, so these 61 brain cell type marker genes, they have a total of 176 alternative transcripts. Some of them are brain specific, some of them are testis specific. Now, there are a couple of papers which have identified such a pattern. So this kind of highlights and brings out that there is a complex tissue-specific and cell type-specific gene regulation going on, and there's a need to study it at the isoform level. So that brings me to the summary and future directions. So TransTEx is able to show that we have about 9% of the total transcripts, both protein coding and non-coding, which are tissue-specific, and testes has the highest number of them. There is prevalence of link RNAs, and that increases in expression classes with either fewer tissues or single tissue versus protein coding genes, which kind of show a reverse trend. Tissue preference can accurately describe the promoter activity in multi-transcript and multi-tissue specific genes. And the brain-specific transcripts are dysregulated in complex brain disorders like epilepsy in the pathway chart, and enriched for non-neuronal and neuronal cell types. And as per annotations, these brain cell type marker genes encode alternative transcripts, which could be brain-specific or testis-specific. We want to extend this to other top cell type, top tissues like liver and pituitary, for example. And also the future directions is to explore tissue-specific relevance of cell type markers of different tissues, look at the cancer relevance, understand what variants are involved, and also understand the evolutionary pattern across different species. So we have this database out there, which is a beta version, where it's a searchable database available freely. So you could either search for a gene or transcript, or you could search for a tissue of your choice. For example, I chose brain here, and then we have the default cutoffs that we used. If you want to use all the samples in GTEx without filtering for their RNA integrity values, you could also do that. And we have some visualizations like box plots for expression values and their exon intron diagram. So the paper, we've gotten reviews for the paper. We submitted it to bioinformatics. So it's going to be out soon, hopefully in a couple of months. So I'm grateful to my entire lab, although half the people are missing in this picture, and my professor, who is a statistician by training, so that helps a lot in thinking. And I'm also grateful to the NSF Travel Fellowship and to Sean, Danny, and to invite me here. Thank you so much. Yeah, and also I'll be looking for postdoc roles. So if you like what I'm doing, please talk to me. Thank you.
Okay, I have one. So you showed that the tissues that had the most tissue-specific things were testis, which is expected. But the second one that people usually get is brain.
Yeah.
But you got liver. So I was wondering if you had any thoughts about that.
Yeah. So actually, in the analysis before, here we filtered out some tissues with very few samples, less than I think 30 samples in GTEx. So before we did get brain as the highest number, but brain and liver kind of compete in that analysis. So some tissue-specific genes, papers where they use tau score, they find brain. So the top three are usually brain, liver, and testes. So these numbers will change if you change the cutoff. So if you think about it differently, you could have different numbers.
So this is a question from somebody outside the field. Mm-hmm. Do I remember correctly that GTEC is from cadavers?
Yes.
So could it be that the reason you and everybody else see brains, liver and testes mm-hmm. is because that's, you have less, it settles down faster in other tissues after death?
That's possible. That's possible. I mean, I was asking about this question, but that's kind of like the limitation of the data. But we could extend this scoring method to like, if there's a specific data set, you could use a similar approach, maybe change the cutoffs a little bit depending on how the probability distribution looks like.
It seems like a great sanity check for your system to take some, if there are data sets available from living people.
Yeah.
I mean, brain would be hard to get, but
yeah,
other tissues.
Yeah, we could
just partition it by PMI and see if the results change if I use like highest PMI versus lowest PMI. Has anybody done that?
I don't think so. I haven't come across it, but maybe there are studies out there and I didn't read them. So yeah, we could also try it with model organisms. I think there are some data sets out there, so I can validate on them.
Yeah,
that's helpful. Thank you. Anything else?
Yeah. Thank you.